Hello, my name's Alyssa Ramirez. My partner is Alexis Hayes, and today we're going to be covering section 10.5, the M phase, chromosome segregation, and the division of cytoplasmic contents. Just to give you all a brief overview of the learning outcomes, we are going to be discussing the five phases of mitosis, the importance of the metaphase, and the differences in cytokinesis of plants and animal cells. So, we know that mitosis is one of the most dramatic and beautifully biological processes that we can easily observe. So, the book really has been able to provide us with some great illustration of each of the steps from prophase all through telophase. So, we're able to clearly see diagrams, and we know that over time and the improvement of the highly powered electron microscopes, we're able to see um, each of the steps clearly, which is pretty cool. Very cool process in science. So, beginning with prophase, which is our very first step of mitosis, you're able to see the mitotic spindle form. So, happening right outside of the nucleus, you've got the mitotic spindle that begins to form out of your centrioles. So, you'll see those little green branches that are kind of coming outward towards the centrioles, and those are also known as it's just like a radial array, and is also called an aster. Okay, so um, from G2 phase, we know that in G2 phase, we had chromosomes that began to condense, and then they just continue to do this in phase stage one of mitosis, and you're going to be able to, they're going to be more evident. You're going to be able to see it now. And so chromosomes appear as sister chromatids that are held together by centromeres. So that's what you see inside of the nucleus. You see your chromosomes, and those centromeres are what are holding the chromosomes together. Those little yellow specks there in the center of the chromosomes. Okay, so in prophase too, you also have your cytoskeleton that is disassembled. So we know that our cytoskeleton was that network that held all of our organelles in place. At this point, it's going to disassemble. And what's going to happen to your Golgi apparatus and your endoplasmic reticulum? They're just going to disperse. And also, you'll have the breakdown of your nuclear envelope. So all these little steps must occur in order for mitosis to continue. And so that leads us to prometaphase. So in prometaphase, now you'll see that mitotic spindle kind of stretch apart. So you're going to have them at opposite ends. So chromosomes attach to microtubules of the spindle at the kinetochores. So we know there's a little point in the chromosome called the centromere. Well, it's going to attach to kinetochores, and it's basically like those chromosomes are kind of placed through kind of like a bead in a necklace. Or we could think of it as like green yarn. We're going to slide it inside the yarn, and you'll be able to move those chromosomes through it. But we know what the potential outcome is, basically, or the goal is for those chromosomes to eventually just line up. And so that's what they're preparing. So the orientation aligns, so kinetochores of sister chromatids are attached to the microtubules from opposite poles. So they are eventually going to line up, move towards the equator, which is the center of the cell, um, by spindle attachment and motor proteins and assembly and disassembly of microtubules. So what's happening is your motor proteins are also being assembled and disassembled. They're going to assemble and disassemble microtubules. So that is what happens in prometaphase. And that leads us to metaphase, where then you'll eventually see all those chromosomes align towards the middle, which is called the metaphase plate. So that is the halfway point between the two poles. So again, there's something that's causing them to do that, and it's the action of the microtubules. So the microtubules cause all that to occur. And so chromosomes are attached to opposite poles and are under tension. So as those two poles are pulling, there's a lot of tension in place. And eventually that's going to lead to them being pulled apart. And so that occurs in anaphase. So anaphase comes around, and you've got all that tension from each opposite pole, and eventually the centromere will. Okay, so what's going to happen is cohesin proteins are what are holding those centromeres together, and they're going to degrade and they're going to just be pulled apart. Okay, by the way, anaphase is the shortest phase of mitosis. So, again, once those proteins that are holding together the centromeres kind of degrade, they're going to just pull apart, and those individual chromosomes become free from sister chromatids. So, chromosomes are pulled to opposite poles as the microtubules shorten, as tubulin subunits are removed from the kinetoshort ends. And so that's all part of anaphase A. And anaphase B is when the spindle poles move apart as spindle fibers slide past each other. So once they're past each other, that's your second part of anaphase. So we're seeing that division now in anaphase, which leads us to the very last stage of mitosis, the telophase. And so here you're going to have your chromosomes that are clustered at opposite ends. You kind of see that there's like a little haze right here at each end. So at each pole, you'll see that little purple haze. And that's just your nucleus that is reforming. So, of course, once we're separating this cell, that's eventually going to happen, the separation of completely of the cell. But as that's happening, there are things that need to occur in each separate cell for you to have one, right? So for the final stage, you're going to have that nucleus reform. You're also going to have the reformation of your Golgi apparatus and your endoplasmic reticulum. Okay. And so chromosomes decondense also, so because they do need to allow for gene expression. Okay, and lastly, you do need to have your spindles and microtubules to break down. So you're going to have the disassembly and the breakdown of your spindle and microtubules. But again, the reformation of the organelles that I just mentioned. Okay, so um, I listed the learning outcomes at the start of this PowerPoint. So I'm going to briefly go over the learning outcomes, which one was the metaphase importance. And so again, positioning of chromosomes ensures that daughter cells receive complete identical sets of chromosomes. So it's not just for nothing. We have these chromosomes that are going to line up in the middle, in the equator of the cell, and the whole purpose is to ensure that each daughter cell receives the complete identical sets of chromosomes. And because remember, when they divide, then that's exactly what happens. Each get one set. And so this ensures the cell is ready to divide, and it also is the checkpoint of cell division. And so cell division is also known as cytokinesis. And that just shows you that you have two equal halves of a cell. And so cytokinesis occurs right after all the phases of mitosis are complete. And we did say that cytokinesis happens differently in animal cells versus plant cells. So in animal cells, we use actin, and actin is just what's going to pinch off the daughter cells. So you've got actin filaments that slide past each other, and the cell pinches and creates what is called a cleavage furrow. So right here in the center, that little crack there is called your cleavage furrow. And then eventually it's going to slice into the cell, making two new cells. In plants, they do not use actin. And again, plants have a very rigid cell wall, it's very thick. It's very hard for actin to work and to be successful, um, to separate the cell. So instead, it is a cell plate that is used in plant cells. So a cell plate is just a membrane component assembled at right angles to the spindle apparatus in the interior. And it grows outward until reaching the interior surface of the plasma membrane, then it fuses. Okay, and then the cell wall is filled with pectins called middle lamella between two new cell walls. So that's the difference between the plant and the animal cell. However, that's a little different in some fungi and some protists. So in fungi and protists, um, some of them do undergo cytokinesis, and all that's happening is your daughter nuclei are separated during cytokinesis, and mitosis occurs not in the outside but in the nucleus instead. So just a recap of what our learning outcomes were, we learned about all the phases of mitosis, the importance of metaphase, and remember the main importance is exactly like why we need to have this phase was just to make sure to ensure that you have two identical daughter cells on each end. And then your animal cells use actin while your plant cells use cell plate. And this is our works cited page. We used graphics from our textbook. And thank you for your attention. Have a great day.